processing, presentation, and finally recognition by T cells. Okay, what are the learning objectives? At the end of this discussion, you should be able to identify, or rather explain the locations and the functions of the MHC molecules. You also need to identify and explain the antigen processing. Mentioning the objectives. Just a minute. Objectives of this discussion. I talked about knowing and explaining the location and functions of the MHC molecules, identify and explain antigen processing and presentation, identify and explain the antigen recognition and T cell selection. You also need to understand the characteristics of T-dependent and the T-independent antigens. That includes the superantigens. And also you can compare and contrast the similarities and differences of T-dependent and T-independent antigens. Now, as you all may be aware, the antigen is any molecule or matter that is considered as foreign by the immune system, and this triggers an immune response. But before this immune response takes into action, this antigen has to be recognized by the immune system, by the various arms of the immune system. We talk about the innate immune mechanisms or the innate immune cells, and also the specific immune response, and that is mediated by either T cells or B cells. Now, antigen recognition by T cells requires presentation by an MHC molecule to the cell membrane or on the surface of the cell membrane, and that is referred to as MHC restriction, meaning an antigen, once it is internalized by the antigen presenting cells, in this case we can talk about the macrophages, dendritic cells, or the B cells. Sometimes the macrophages are referred as monocytes when they are in blood circulation, or macrophages when they are fixed in the connective tissues. Then this antigen has to be broken down in similar molecules, what we talk about the peptides, and then from there they are linked with special proteins, which we talk about later, and transported to this MHC molecule. Now, this MHC is a group of special proteins coming from the major histocompatibility complex genes that are specialized in antigen presentation, and this is the target for recognition by the T cells. So because T cells require or rather seize or responds to an antigen that is coupled with an MHC, or what we say as antigen MHC complex, then T cells are said to be MHC restricted, and we'll see that later. Now, there are various pathways for antigen presentation, and there are two of them. We have class 1 MHC associated with peptides from within the cell, or what we call endogenous antigens. And one classical example, you remember the viral or the viral infections, mostly all the viruses are obligate intracellular pathogens, meaning they must enter inside the cell, utilize the host machinery for replication, and then come out in large numbers as antigens or as virions for infecting or for circulating, and this can be recognized by the immune cell. So while inside the cell or the target cell, this virus will generate foreign molecules or proteins or antigens, and this will be recognized within the internal mechanism of the cell. And that involves breaking those antigens into smaller pieces, coupling them with an MHC molecule, and then this MHC goes some changes that finally this antigen MHC complex is finally exposed onto the cell surface or the cell membrane, and this will be targeted by T cells, depending on which nature of the MHC that is involved. If it's MHC class 1, then it becomes a target by the CD8 cells, or if it's class 2 MHC in association with peptides, then it becomes a target by the T helper cells. Okay, so we have these two pathways, class 1 MHC pathway, sometimes you can call it endogenous pathway, or class 2 pathway, which we can call exogenous pathway. Now, this diagram shows you the earlier discovery about these MHC molecules, and an experiment was done just to depict that these antigens are specific and the T cells that are involved are different depending on the nature of MHC that is involved in presentation. So if you have the LCM virus, it can always be an intracellular pathogen, and that can be presented by the CD8 or cytotoxic T cells. But if you have an antigen that is not intracellular, this can be recognized by the T helper cells. So those are some of the early experiments that people utilize just to find out how these antigens can cause stimulation of T cells whenever they are coupled with an MHC molecule. And remember, the antigen presenting cells, they are the ones that they process the antigen and couple it with their own MHC molecule, and then this becomes a target for recruitment by the T cells. That is shown in this experiment is just to indicate that the mouse was infected with LCM virus, and this, you can have the spleen that is containing T cells, and this sometimes can be recognized by the CD8 cells, which can release certain molecules in form of granzymes and bufferin, and this will end up creating holes or poking holes on the target cell. And the work of the T cells is not to prevent infection, but rather to deal with infected cells. So like now, what is happening, we are talking about the COVID-19 pandemic. The SARS-CoV-2, yes, it can infect, but you need a vaccine to boost the immune response by activating the T cells, which can generate memory, and that can be important in reducing the severity of the disease as what is happening with the current vaccine. They cannot stop infection, but having that they generate memory for T cells to recognize the same antigen, then there's more machinery in terms of immune response as opposed to somebody who is not vaccinated. So I'm just passing the message that you need to be vaccinated, or you can advise somebody to be vaccinated irrespective of what is going on around. Anyway, let's continue. Now, there are various distinctions between MHC 1 and MHC class 2. Now, if you look at MHC class 1, most of the cells or the target cells can present an antigen with an MHC molecule to T cells, specifically the CD8 or cytotoxic T cells. But in case of MHC, only an antigen presenting cell can present an antigen with an MHC to T helper cell. Now, I give you an example. Somebody who has hepatitis viral infection. Your hepatitis organ or the liver has the specific cells or hepatic cells. And whenever you get infected with hepatitis virus, the cells are infected. And these cells, they also have the MHC class 1. Then this MHC class 1, with the help of the proteosomes or enzymes that cleave those hepatitis antigens, they can present it along with MHC to the cell surface. So that becomes a target by the CD8. So sometimes whenever you have people with intracellular pathogens, first they can be recognized by innate immune cells like the natural killer cells, which try to target the virally infected cells by monitoring the MHC expression on their surfaces. So whenever there is minimal or no expression of an MHC, then natural killer cells would utilize either the killer activating receptors or inhibitory receptors if there is no infection, and those receptors are very important. So failure from the natural killer cells, there is another arm of specific immunity that is provided by the presence of the CD8 cells. So like what happens sometimes in hepatitis infection or chronic infections, the much of the damage that is done is not due to the virus really, it's because of the excessive or hypersensitivity due to CD8 responses, because there's a lot of chemicals that are released, and those are the ones that cause exaggerated or hypersensitivity reactions for that. Now, in terms of MHC class 1, we talk about that which are, they have this MHC class 1. So apart from the professional antigen presenting cells, all the nucleated cells have the MHC class 1. And I've said they can be exposed to viral infection or any intra cellular pathogen. You know, we have other pathogens that can still infect the cells, like the microbacterium. There are many others, and we have also the other pathogens that cannot necessarily enter the cell, but they can remain on the surface. If you remember, like somebody who is having the normal flora that prevents or blocks from other infection by competing with the available resources in terms of nutrients. But should you get cut or injury to the skin, sometimes some of these normal flora can be turned out to be pathogenic. So the staphylococcus, the streptococcus, those bacterial infections, they fall under those external or exogenous antigens, okay? Now, in terms of antigen presenting cells, they are categorized into two categories. We have professional antigen presenting cells, and we have non-professional antigen presenting cells. So the professional can do two things. They can phagocytose or engulf the target antigens, then couple these antigens with an MHC molecule, and then present them to the cell surface for recognition by the T cells. Depending on if it's MHC class 2, obviously that becomes a target for the T helper cells. Now, non-professional antigen presenting cells, these are other cells like the Langerhans or some special epithelial cells of the skin, which can play a role of presenting an antigen even though they are not professional phagocytic cells. You have even epithelial epithelioid cells on the skin. You remember, like somebody who is exposed with maybe poison ivy or some solutions in maybe some soaps, they can have these haptens, which are small proteins but not antigenic on their own. But when they mix up with globulin, maybe through the skin, they can expedite or elicit hypersensitivity reaction. And in that way, you find like the skin behaved like an antigen presenting cell, even though that is not a professional antigen presenting. We have also some vasculature or some endothelial cells that also can present antigens, but not necessarily being phagocytotic in that nature. Then in MHC class 1, we have the association between MHC class 1 and an antigen. And this becomes very conspicuous, especially with cytotoxic T cell mediated immunity, as opposed to MHC class 2, which is coupled with a humoral immune response or antigen, and not really antigen, but antibody generation, because T helper cells can only activate B cells to produce antibodies. But sometimes the B cells can be activated to go into plasma cell, which can remain without necessarily generating the antibodies. So these are some of the differences in terms of MHC class 1 and MHC class 2. The key points are that what are the targets and in terms of antigen and MHC association, and which cells will recognize these complexes. Is it T helper cells or the T cytotoxic T cells? Now let's look at the antigen processing through two separate pathways that I've mentioned. Now, the first pathway we can talk about cytosolic pathway. There's another name we have discussed previously, endogenous pathway, and this can sometimes be called class 1 pathway. And what happens endogenously, the antigen is maybe phagocytosed or broken down by the proteosomes, which are hydrolytic enzymes, and this will generate smaller peptides that can be coupled with transporter associated proteins, which can transfer these small peptides to endoplasmic reticulum, and then this is linked up with MHC molecule, and that becomes an MHC antigen complex, and that will be recognized by the CD8, because CD8 cells only recognizes an antigen in association with MHC class 1. Now, the other pathway we just can mention is about endocytic pathway. Now, why do we talk about endocytic? Remember, antigen presenting cells are the phagocytic cells. We talk about the dendritic cells, the monocytes, stroke macrophages, even the B cells. But the B cells are not professional phagocytic cells, even though they can present an antigen, because they use their receptors. You remember the B cells have the immunoglobulins on their surface, which they utilize as receptors. So when they bind, they just make like a cleft or a Clathrin receptor-mediated endocytosis. So they kind of curve towards the antigen, but they don't fully engulf the antigen. But dendritic cells, monocytes, stroke macrophages, they are very good phagocytic cells, and now they have MHC class 2. When these antigens that have been phagocytosed or endocytosed, they become broken down. They are now linked with an MHC class 2, and this becomes a target by T helper cells. So endocytosis, remember, there is engulfing and making endosomes. So sometimes when you have endosomes that is linked with lysosomes, you form the phagolysosome, and this is what we are talking about. If you have this classical example of this kind of mechanism, you look at, for example, mycobacterium infections, because these are the pathogens that can be, they can infect the antigen-presenting cells, the macrophages, and this can trigger continuous immune response, especially in the lungs, because there is continuous activation of T helper cells, and depending on the nature of these antigens, sometimes you don't have antibodies that are triggered, that are very important in eliminating this immunity. And that's why in the case of TB infections or mycobacterium tuberculosis infection, you only have to generate a T cell memory rather than antibody, because antibodies don't really work. Especially even with the COVID, you have had like the vaccine that generates only the antibodies don't work, but you need to generate memory in terms of T cells. So that is endocytic pathway, and we are going to see further examples. Now, the processing of an antigen, endogenous antigen, that is the cytosolic. Why do we call it cytosolic. You remember, within cell, you have the nucleus. Outside the nucleus, you have what? Cytoplasm, and then you have the covering or the cell membrane. So when we talk about cytosolic, in reference to the cytosol, and this is a liquid state of the cell that shields the cell from many other things. So the cytosol provides a very good room for the pathogens, especially the viral infections, to replicate and make several copies. And that is why it drives the name cytosolic pathway. Now, this processing of endogenous antigen involves three activities. Number one, peptide generation from proteolysis, or the breakdown of peptides into smaller peptides. So it's like cutting. If you go to Mumia Sugar Company, you look at the way sugar is obtained from the shamba or from the farm, and it's taken to different stages. But there is the initial stage of cutting those sugarcane into smaller fragments for able to go to the next level. So just the same way, an antigen cannot be recognized as it is unless it is broken down, because even this requirement is directed by the MHC molecule, because it has specific amino acid length of peptide that it can recognize or bind to, right? Then another, after breaking down, you need to transport, or these proteins in form of peptides need to be transported. So another step is to transport these proteins or peptides to endoplasmic reticulum that is involved in this process. Then you have lastly, there is the binding of the peptide to an MHC molecule. So that is the three steps or activities that happens for this particular processing. Now somebody will ask, what if this process does not take place? Don't expect any immune response. If it's a T cell, immune response, this will not happen, because this process of breaking down is critical, because it will create smaller portions of peptide that can be recognized or bound with an MHC molecule, and that becomes a target for T cell recognition. Now this slide shows you more details on what we have just discussed. Now we have a peptide that is shown, or an antigen, and this is coupled with a transport molecule which is known as ubiquitin. This does the process, the transport whenever it's coupled with this antigen, and this will be further moved to the endoplasmic reticulum. Of course, this requires some level of energy, and that's why you have the involvement of ATP. So the proteins targeted for lysis will combine with this molecule we have talked about, ubiquitin, and that is the role. It enables these smaller peptides to be moved or to be transported to the next compartment. Now this ubiquitin protein complex is degraded by proteasome, and that is what I was saying, like unless you have this antigen or protein broken down, it cannot be bound with an MHC molecule. So you can see the critical role that ubiquitin plays in terms of binding with this complex, and then the next level is to have the proteasomes. These are special enzymes within the cell that does the breaking down, and that becomes very critical in generating smaller peptides. If you have done MHC study, it has different lengths of amino acid peptide that it can recognize. I remember MHC class 1 requires amino acid peptide length of between 8 to 10 amino acids. But when you talk about MHC class 2, some book says from 13 to 20, on average 18 amino acid peptide length. So that becomes different sizes in terms of what is required to fit the groove of the MHC molecule. Now let's look at the endogenous processing further of this antigen. So the peptides that have already been generated from the proteolysis process, they are bound to the transporter proteins associated with antigen processing. And this we call them the TAP. So we have TAP1 and TAP2. Their work is just to transport whatever that is already broken from the previous stage. And this TAP is heterodimer, which is using the ATP as a source of energy to transport these peptides. And I said earlier on that they are between 8 to 10 amino acid length. And this is transported to the lumen of the endoplasmic reticulum. And this is very, very, very critical. Usually the basic amino acid at the carboxyl group, that is the C or H end of the peptides, is utilized for this process. So remember the TAP1 and the TAP2 when it comes to endogenous antigen processing. Another name for endogenous we have talked is class 1 pathway, and another name we have just talked is cytosolic pathway. Now let's look again what happens in case of an infection on a protein. As the diagram is showing, you'll have the generation of maybe foreign antigens or proteins within the cytosol, as the diagram indicates. Then this will be broken down into smaller peptides, and the work is done by proteasomes. And this will be transported by the TAP1 and TAP2. Finally, it will reach the destination, which is the rough endoplasmic reticulum, and there they will be coupled with an MHC molecule. And this one will become a target for recognition by the T cells. And that's why in books, they talk about that T cells are MHC restricted, meaning they can only see antigen MHC complex. Depending on MHC class 1, it becomes a target by CD8 cells, or if it's an MHC class 2, becomes a target by the T helper cells. Now this diagram, or rather the figure, shows you finer details about other molecules that are very critical in this MHC complex in terms of stabilization. So we have the calnex, which is very critical in stabilizing the MHC class 1. And this, we have also the beta-2 microglobulin, which also binds, and then this enables further release of the peptides that can be coupled with MHC class 1. So this is a classical example of an intracellular infection. Okay. Now let's look at the other cycle. We can talk about class 2 pathway. We can talk about exogenous pathway or endocytic pathway. So there are 3 names for this class, this pathway. We talk about class 2. You can mention about exogenous because the antigen is totally coming from outside. It's not replicated within the cell mechanism. And another name is endocytic because these antigens are phagocytosed and they form endocytic endosomes when they are phagocytosed or engulfed by the phagocytic cells. Okay, so these exogenous antigens are typically phagocytosed, which I've mentioned, or endocytosed by either macrophages or other monocytes, and also other antigen-presenting cells, not to forget about the dendritic cells or the B cells. The foreign antigen is degraded within that phagolysosome or that fascicle that is formed, and this sometimes is expedited by the vacuoles that are found in these antigen-presenting cells that are rich in certain enzymes. We call them lysosomes. So these enzymes are very critical because they work at certain pH, and also the pH helps this breakdown of engulfed material. The antigen is degraded into 13 to 18 amino acids, which I mentioned. You can see now there's a difference. While the MHC class 1, we were talking about 8 to 10 amino acids, but in this case, we are talking about 13 to 18. In some books, they talk about 20 amino acids that are generated, and this is the right fit to feed the group of MHC class 2. So the peptide length is very, very critical, because if it is below, maybe it's not so much a problematic, but when it is big in size, it may not completely fit. So sometimes the best or close to the right size gives you the best or the optimal result in terms of binding of the MHC molecule. Then eventually the endocytic vacuole returns to previous condition, and that becomes recycled in many cycles, because these cells that participate, the antigen presenting cells, once they have done one presentation, they can still go back and do another antigen. They don't necessarily have to die every time that they present an antigen. Now, if we look at this processing of external or exogenous antigens, remember these are not intracellular pathogens, but rather like exogenous, in the context that they are not generated within the cell. So don't confuse when we talk about exogenous and endogenous pathogens. Now, the cycle, we have said it's endocytic because, as you can see, you get an antigen, it is engulfed, and this is an classical example of the B cell, because the B cells have the B cell receptors, which are immunoglobulin, and they just bind an antigen, or sometimes cause a false phagocytotic process. And what happens is when you see these creatine-coated vesicles, all these invaginations of an antigen with the receptors, it kind of activates the release of the expression of an MHC molecule, which now coupled this particular antigen with the help of the pH that is between 5 and 6, and then these lysosomes, which are also very important enzymes that can break down that antigen, and it releases those smaller peptides between 13 and 18, and this becomes attached with MHC molecule, and finally it's released. If you are very keen, you can see that the MHC is expressed beyond the pH stage. That means sometimes you don't have the MHC already expressed when the cell is not activated. I mean, when the cell is not yet interacted with an antigen, the MHC proteins, the genes are present, but the proteins are not yet expressed. But whenever you have this activation, there are various signals that are sent to the cell, and then the genes are translated into proteins, and these proteins are now the MHC we are talking about. Then the MHC will find itself coupled with that antigen, and it will come to the cell surface. Once outside the cell surface, then the T cells will recognize. And in particular, for this case, it's T helper cells, because we are talking about class 2 MHC molecule in association with an antigen. Now, processing of antigen or exogenous antigens involves various different molecules as opposed to TAP ubiquitin. But here we are talking about different proteins. We talk about the invariant chain, abbreviated as LI, which is very critical in coupling these antigen and finally presenting it. So with the invariant chain endoplasmic reticulum, the alpha and beta genes of MHC class 2 combine with a protein, and this invariant chain is expressed as CD for 74. I'm sure you have learned about the cluster differentiation, which is a systematic way of identifying certain biological markers that are found on certain cells. Now, the invariant chain binds to MHC molecule at the peptide binding cleft and then exits the endoplasmic reticulum to Golgi apparatus. As proteolytic activities continues, this is contained within the lysosomes. The invariant chain is degraded to smaller fragment, which we call clip, and then the cycle becomes repeated now and again. So what to note here is that MHC class 2, you don't need the TAP1 and the TAP2 or ubiquitin, but instead you need this invariant chain molecule that is very critical to coupling an MHC also with an antigen. And here, remember the enzymes here, we are talking about the lysosomes as opposed with class 1 MHC or class 1 pathway, where we are talking about proteasomes. So if you want to describe the exogenous pathway, you can describe in the context of class 2 MHC, invariant chain, and finally, the release of the clear peptides, which are very critical for achieving this process. But in case of class 1 pathway or endogenous pathway or cytosolic pathway, as maybe what you can remember first, we are talking about the proteolytic enzymes which break down. Then you talk about the TAP proteins, which are transporter associated with antigen processing. And then finally, you have the expression of and stabilization of MHC class 1. So those are the two critical differences that you can remember. And of course, MHC class 1 will be recognized by the CD8 and MHC class 2 by the CD4 cells or T helper cells whenever in association with antigens. In comparison of the antigen processing pathways, we have just mentioned about the two, but you can have a look. Endogenous pathway, we have talked about the proteasomes as proteolytic enzymes that break down. Then the molecules or the peptides are linked up with transporter associated with antigen processing, that is the TAP1 and the 2. And this one is taken to the endoplasmic reticulum, and finally, this is released and all other recognized and or activates the generation of MHC molecule. While in class 2 or exogenous pathway, we talk about invariant chain, and then you have no much of these proteolytic enzymes or proteasomes, and you have the linkage with class 2 MHC molecule. So the process in terms of exiting the MHC is clearly indicated as we have described. So I think that is one is well understood. Okay. Now, in terms of just trying to instill the same points in form of a question that I've posted there, what does the alpha beta T cell receptors recognize? Or rather, how is the T cells activated? We have talked about the fragments of proteins that are broken down and associated with an MHC molecule onto the cell surface, and that becomes attacked by the T cells. But remember which type of MHC that you are talking about and what type of the T cells that will be activated. Helper T cells recognize peptides in association with MHC class 2 molecules. What about the CD8 cells? I think you can even guess they are important in recognizing an MHC that has been associated with class 1 molecules. Now, briefly, let me talk about the superantigens. We have seen that in the true immunological response, whenever you have an antigen, this antigen has to be recognized or processed by antigen presenting cell, and this becomes available in form of an MHC antigen complex for recognition by the T cells. But what happens in the case of super antigens? These antigens never require to be processed and presented by the antigen presenting cells to the T cells, but rather the intact protein or antigen binds to the variable region of the beta gene of the T cell directly, and it has antigen presenting cells. So it's more of a direct way to reach the T cells by interacting with the T cell receptors, as opposed to being coupled with an MHC molecule on the start. So that is what happens with the super antigen. And these examples of super antigens you can read in books. They are like exotoxins or endotoxins that are produced by certain bacteria, and sometimes they can be very lethal. And that's why we talk about in vaccines, where you want to detoxify these toxins. We talk about toxoid. So you can have these antigens and make them less harmful by treating them chemically, and becomes less toxic, but they can induce immune response in form of vaccines. Okay. Now these super antigens, we have talked about the very origin of T cells, and they can activate large number of T cells, which can release so much of the cytokines. And this, whenever you have a storm of or a cytokine storm, for that reason, you can have hypersensitivity reaction, which are associated with pathological effects and sometimes even anaphylactic shock. So this is what happens, like in case of COVID, you have had some people with those who present with severe disease, they have the cytokine storm, meaning there is too much of activation with cytokines, and this causes pathological issues for these particular patients. And I think that is the end of this discussion. You can still get more information from the textbooks. This is not the end, but I believe I've given you enough guideline that you need as you do your personal studies. I want to stop there. Anybody with a quick question? I don't want to close you out. All of you are okay? Okay, if there's no question,